think anytime I see specifically something probably related to parental care in amphibians is like the birth of one of these tiny marsupial frogs is a special moment that I would say those are very unique moments.
My guest today is Dr. Alessandro Catenazzi, Half-Earth Chair of the E.O. Wilson Biodiversity Foundation's principal program, the Half-Earth Project, and also Associate Professor of Biological Sciences at Florida International University. It's a pleasure to have you.
That's my pleasure.
So let's talk about Half-Earth first. Can you tell me what the Half-Earth Project is?
Yes, the Half-Earth basically is a project that implements Ed Wilson's vision about basically trying to protect as much of the landscape, both terrestrial and aquatic, as a way to preserve biodiversity. And the idea is why Half-Earth is because it's a simple concept. Is half of it. Everybody relates to half, right? And is that enough? Is that not enough? That's kind of beyond the point. Is so that we have perhaps a very ambitious goal, but we do have a goal, and the goal is to be able to set aside enough nature so that we can preserve biodiversity, and we're part of it, and we also depend on that biodiversity to be able to thrive as a species.
And how do you determine what half to try to preserve then?
Yeah, I think for that, part of the mission of the foundation is to really promote all these activities that increase our knowledge about where biodiversity is distributed. I think obviously there's a little bit of a utilitarian perspective from that, but we are in a crisis, and so we really need to know where the highest priorities are in terms of protecting biodiversity. And we know that biodiversity is by means now equally distributed around the globe. We have places that have much higher diversity, and we also have places where endemic species, highly restricted species, are highly concentrated. And that's also often the places where they have already gone extinct. And so if we look at many groups of animal and plants, where we see a concentration of extinct species is typically on islands, right? Because islands were shielded from predation, etc., before humans settled them. And also organisms there, they were able to have their own evolutionary lineage, very distinct from organisms on the mainland. So those are, if you think about amphibians, the group I study, for example, the ones that are best documented extinctions are typically of island organisms, but the same for plants and other animals as well. Also, it helps that they are very concentrated environment, very tightly defined environments, the islands, and so it's easy for us to say the species has gone extinct here, as opposed to in the Amazon forest, which is a much bigger place, and you can always be sure if something is there. We can never be sure 100% that a species has gone extinct, but that's possible on islands. And so for much bigger landscapes, that information on where we can find the biodiversity and how we can best manage it, because at this point, really, I think for most of the planet, we are looking at a very active role in managing that biodiversity to preserve it. Otherwise, we have all these emergent threats that make it impossible to just kind of let it aside and hope for the best, because we have climate change and species are going to migrate, and so we have to be able to set up, for example, kind of corridors or protected areas where those species are able to move and find suitable habitats. And so I think the foundation, there's this Map of Life project, which specifically aims at making that biodiversity information. A lot of it is already there, but it's just perhaps in a form that's not so easy to use. And that's the Map of Life project that collaborates with the E.O. Wilson Biodiversity Foundation. And then the Half-Earth project specifically has this program with the Half-Earth chair and scholars. And so embedded in that, there's a mentorship program. And so the chair is a mentor, and currently there is a single chair, but the idea is to have multiple chairs. And there are two scholars, two Half-Earth scholars. And typically, at least in my case, but also with the previous chair, there's been, I think, this idea that people that are involved in biodiversity research and taxonomy and discovering new species, and also transmitting that knowledge, that know-how of how you go, how you are best able to document new and discover new species and document that biodiversity to the next generation. And so there's a strong mentorship component to that program.
In the Scholars Program, these are students around the world or in only specific areas?
Yeah, I will say, I mean, there's only been 2 chairs, right? I'm the second one. So I don't know what the long-term tradition for the foundation will be, but so far there's always been a strong connection to place. And so the previous chair was working in Africa, and so there was a strong connection there. And I work in the Andes, in Peru specifically, and so both scholars also work in that region, especially the Amazonian Andes, which is a place that for biodiversity is extremely important for a wide variety of organisms, because we have incredibly high endemism. And so basically each valley, each mountain pass has its own set of species, especially on the Amazonian side.
And how many scholars are taken into the program every year?
Currently we have 2.
Is it 2 every year then?
I think that has varied over the years, yes, but currently we have 2 scholars.
And you mentioned high biodiversity. Could you talk a little bit about why we have such high biodiversity in the Amazon?
Yeah, I think for many groups we have biodiversity generally concentrated in tropical areas, and there's many different ideas about that have tried to address this idea of why we have more species in the tropics, and some of those ideas have to do with species-zero relationship. And if you properly project, right, the Earth, you'll see that tropical areas actually are quite vast. You also have this idea that during ice ages, for example, glaciation events, especially in the temperate areas, you had climates that were not conducive to life. And so the evolutionary history of organisms is automatically much longer in tropical areas. That allows for greater speciation. And there is also, of course, the idea of direct climatic effects, and so warmer temperatures, which will also promote higher speciation rates, and also the absence of seasonality, which also allows organisms to better specialize to specific niches. And so this idea, Dan Jansen, this tropical biologist, that mountain passes, for example, are higher in the tropics, right? And so the tolerance to, for example, different climates for a temperate organism is much wider, which means that temperate organisms can actually be widely distributed, which means gene flow, which means less species, whereas in the tropics, species have climatic niches that are much narrower, because over evolutionary time, they basically have had a pretty stable environment. And basically, as you go up a mountain, for example, the turnover, as you go up the mountain, every 200, 300 meters of elevation, you have a much faster turnover in the species composition of those communities. Those are just some of the ideas.
Yeah. Could you talk a little bit about how the amphibians change as you go up in elevation in the Andes?
Yeah, with amphibians, so amphibians are actually really interesting. Amphibians is this class, it's got 3 orders, Caecilians, which are strictly tropical. Then we actually not native to the United States, but now we have an invasive, potentially invasive, there's a Caecilian from Colombia that's been introduced to Miami. So they're loose in the canals in Miami, but otherwise tropical distribution for Caecilians. Then frogs is by far the most species-rich order of amphibians. But the third group, salamanders, is actually one of the exceptions, going back to the idea of why we find so many species in the Amazon and in the tropics. Salamanders actually do the opposite, and there are more species-rich in temperate areas, and specifically in North America. And
the Smoky Mountains.
Smoky Mountains, yes, the Smoky Mountains, Appalachia, and also Mexico. It's much more species-rich. But for frogs, yes, we see kind of your general patterns where you find just one species, as you know, starting from northern Canada or even central Canada. As you go south, then you steadily increase the species as you approach the equator. And also, let's say if you start from the Amazon and you go up towards the peak of the Andes, you also have a pretty gradual decline in the number of species. And so a place in the lowland Amazon, which is the most species-rich place on Earth for amphibians, but most of them are frogs, can have over 100 species of amphibians, between 100 and 120 species of amphibians. And the very top of the mountain, the highest so far is actually in southern Peru, where people have found frogs up to 5,400 meters, and that's quite high. But even places like the Peruvian-Bolivian plateau, Altiplano, which is between 3,400 and 4,000 meters, you have 3-4 species at those elevations.
Do we have any idea why salamanders are the outlier then for amphibians in terms of diversity being found in temperate regions versus tropical regions?
Yeah, salamanders, the idea is this is a very ancient group, and there's many characteristics that make them very intriguing. And they also have, among vertebrates, kind of messier DNA. Their genome is very large with a lot of seemingly junk DNA, right? And so a little bit, one of the hypotheses is the salamanders are kind of, I don't want to put it too much in a negative light, but it seems like they might be doomed in terms of evolutionary lineage. And so they're kind of attached to the past, and their niche, which is what they really, where they really thrive, is more like colder and highly humid environments. And those you tend to find in places like, well, the Smoky Mountains, right? Mm-hmm. And so it's a group that's now being able to diversify into these much warmer environments. But, that's just one of several ideas. But it's a group that has these marks, especially in the genome, of not being a lineage, evolutionary lineage, that's been as successful as frogs, for example.
Mm-hmm. And do you notice any morphological changes in frogs, for example, as you go up in altitude in the Andes?
Yeah, that's a very interesting question. So there's this idea in biogeography that, although this idea was developed for endotherms, which are mammals and birds, that as you go up in elevation, the organs should be bigger, right? Bigger in size. And that's not necessarily true of amphibians. In fact, although it's a relationship that sometimes you see within species, and as you go up, the individuals within that species, sometimes they are larger as you go up. We do have among the smallest amphibians are actually frogs that have been found at high elevations. Yes, and one of the species I discovered, Noblella pygmea, we named it pygmea for a reason. It's one of the smallest frogs, probably in the top 5 smaller frogs worldwide, and we found it at 3,000 meters. So it's a very high elevation frog, and yet it's extremely tiny. And that group, Noblella, actually, it's quite species rich, from 2,000 and 30. Actually, almost 4,000 meters.
Mm-hmm. And so you've discovered a lot of new species to science. What is that feeling like when you discover a species that is new to science?
Yeah, what I really like is about seeing the diversity of the forms, the coloration, their calls. I really enjoy being able. This actually is those things a little bit different, and also I think the realization that behind any new form, and at first you just see, okay, this is probably new. I'd never seen it with this coloration, with these tubercles, with this specific call, but it's also a whole evolutionary history and behavior, sets of behaviors, natural history. A lot of them, secrete peptides, for example, through their skin. A lot of them have parental care, fascinating parental care. With one of them, for example, we were able to prove in these marsupial brooding frogs that the mother actually transferred nutrients through the skin to the developing embryos. So it's basically the females, they have these pouches, closed pouches, where they carry the embryos, the fertilized eggs. After fertilization, the male helps the female and pushes these eggs inside that pouch on the back. And actually, a herpetologist from Ecuador, she's dedicated her life to these marsupial frogs. And she already wondered in the 80s whether there was this kind of very intimate connection that could also allow for the mother to directly provide nutrients to the embryos. But she didn't really have the means at the time to be able to test it. We did it a few years back. using stable isotopes that allow us to track and basically feeding the mother with heavy isotope that we could then track into the tissues of the embryos. And sure enough, they do transfer, which is basically, it's crazy because it's like the skin acting as a placenta, basically, even though it has nothing to do with a placenta.
So the nutrient transfer is skin to skin.
It's skin, it's skin of the mother through the membrane of the egg, and then it's probably picked up through the gills of the embryo inside the egg. So they don't have tadpoles, but they still have gills. It's just that the embryo is developing inside the egg.
And then the embryo, as it grows, at what point do they leave the pouch?
When they hatch, then they just squeeze out of the pouch, which is really funny to see.
Yeah. A miniature frog, then.
A miniature frog, yes. Yeah. So I think, yes, I think that makes it really exciting to me. Obviously, just discovering something new, you put a name, that's great. I think it's also great because a lot of conservation is based on what kind of species you have, and especially a lot of the species that we discover are endemic species, and often endemic species are a good justification to be able to protect a specific area. And so that's, I think, a very tangible and concrete contributions to conservation.
Another interesting species I was reading about, could you talk about the gladiator tree frog, a little bit about the natural history of that and how they earned that name?
Yes, gladiator tree frog is a group of tree frogs that have these combats, and so the males they fight over territories, over basically who's going to attract the female for mating purposes. And in many species, they have spines on their thumbs, which they use in a fairly aggressive way. There's a species that we have studied quite extensively in Manu National Park, in the cloud forest in southeastern Peru. And there it can even draw blood from our skin. It's happened, not that common, but it does definitely cause wounds when they have these male-to-male combats. It has been a focus of our interest, mostly because it wasn't so much for the behavior, but mostly because we study a lot a fungus, a chytrid fungus, that has been highly destructive to amphibians worldwide. We think its origin is it's in Asia, maybe in the Korean Peninsula, on the basis of genetic data. We don't know how it got out and how it suddenly spread to all continents, basically. But genetic data basically support the idea that it's a recent emergence. And it arrived to this area of the Amazonian Andes, probably sometimes in the early 2000s. And it has caused a dramatic decline in the species of amphibians there. But the species of gladiator frog has been able to persist, not quite thrive, but to persist in areas where all the other frogs that used to be syntopic, that used to live in the same area, they disappeared, and specifically in creeks. And the fungus is aquatic. And so especially toads and tree frogs that were highly dependent, like glass frogs or toads that were highly dependent, harlequin toads that were highly dependent on creeks for reproduction, they basically were extirpated or possibly extinct for some of the species. But these gladiator tree frogs were able to persist. So obviously we wanted to know why they were able to persist in this environment. And that's why we started studying them more in depth.
But we don't know yet. The studies are still ongoing.
No, we do have some of the
do.
answers. So this specific fungus, this mycosis, is cutaneous, is on the skin. It secretes a toxin that actually interferes with the development of lymphocytes. It does the same to human lymphocytes, by the way, but we're protected because we're too hot for the fungus. This fungus likes moderate temperature, 18, 20 degrees Celsius, so it's perfect in the cloud forest. So obviously we had to look in the skin for ways that these tree frogs could defend themselves. And two of the main lines of defense they have, in general amphibians, not just tree frogs. One is the microbiome, the bacteria and yeast and even viruses sometimes that live on the skin, because the skin is always a very moist environment in frogs. So you have hundreds of species living, 100 species of bacteria living on the skin. And so often these bacteria, a lot of them secrete toxins, a lot of them, they are in this constant struggle against each other and against fungi. And so we studied first the microbiome of these tree frogs to be able to understand whether the microbiome had species that were good at killing the fungus or slowing down the growth of the fungus. And sure enough, we found many. And it's kind of hard to tell because we haven't tested really the ability of all these bacteria to slow down the growth of the fungus. But we can we observe that there's a really good association between the presence of specific bacteria and how infected the frogs are. And so that to me is pretty suggestive of the fact that the bacteria play an important role in defending the tree frogs. And so notice that it's not that they don't get infected, they do get infected, but their microbiome helps the tree frog keep the fungus in check, right? And so it's still in the skin, but it's not spreading all over the skin because now there are these bacteria that are really good at some of them might secrete toxin that kill the fungus, some of them they just grow faster, who knows? It's a very complex environment, very dynamic environment.
But you've only found this in the gladiator frogs.
So we have compared the microbiome of the gladiator tree frog with species that occur at the same site. And by far the microbiome of these tree frogs is much better at inhibiting the growth of the fungus than the other frogs that we find in the same environment. So other frogs also have that, but it seems that these tree frogs, by luck, because really that's not something that is necessarily transmitted down across generations because they pick up the bacteria from their environment. So it's hard to know exactly what made it possible for these tree frogs to have just the right mix of bacteria to be able to defend themselves.
Have you witnessed males in combat?
Not the whole, no, not the whole thing. I wished because it will be. I think it will really inspire me to extend studies on the other side of the defense, which is that they also secrete peptides, and most amphibians secrete those skin peptides, and many of those peptides, they have functions that are antimicrobial. Often those peptides, they form kind of a cylinder, and so it's basically a hollow cylinder, right? And that's what pokes a hole through a cell wall of a bacteria and causes all the water to flow into the cell and makes it basically, it explodes, right? And so it's very effective at killing bacteria and fungi too. And so we started studying one of those peptides that we discovered. It's a novel peptide that's not been documented before. It's similar to peptides that have been discovered in other tree frogs. And we then tested, right, the peptide. Once you can basically determine the sequence, which we did by looking both from what I call the upstream and downstream. So the upstream is looking at the genome because these peptides are in the genome of all the frogs. They're in their genetic code. And so we sequenced the genome and the transcriptome to make sure that we could find the sequence for those amino acids, the codons for those amino acids that form that specific peptide. And also downstream, which we can do with mass spectrometry. And so we go out and we actually use a mini taser that it's a small, it's just like a few volts that triggers the release. It doesn't harm the frog, it triggers the sudden release of those peptides from the glands in the skin. And so then we can run a mass spectrometer. And the mass spectrometer, in the old days, it will just give you these MZ ratios, and so peaks that you then had to figure out what they represented. But now with the new machines, you basically get the sequence with the amino acids of the peptide. So once we have the sequence, then we can order the peptide from the lab. They just make it de novo, and then we can have basically clinical tests, right, where we tested the peptide against the fungus. Now, what's very, I think, important for these studies is that we had both the upstream and downstreams, because actually what you see from the transcriptome, which is already a pretty good prediction of what you can expect in the skin, is actually not what you find in the skin. And so that's why it's very important to also look at what you get from the mass spectrometry. Those are the actual fragments that you get on the skin. And it turns out that after the secretion, there is probably enzymes on the skin that cleave that peptide into smaller fragments, and that's what we find in the skin. It's actually smaller fragments. So we tested the whole peptide and we tested the fragments. And it turns out the fragments don't do anything to the fungus, but the whole peptide, which probably is concentrated in those glands, that is actually really good at killing the fungus.
Yeah. So they have quite the fitting name, not only for the combat, but also the immune combat, right?
Yes. So what's interesting, there's another twist in this story is that, and it's kind of a hypothesis here, and that's why I really would like to witness one of those combats, is that most likely, I mean, this is a new novel disease, right? And so it's pretty unlikely that that peptide evolved in response to a very novel disease, because it's not an evolutionary history of this frog. Assuming that it's really a novel disease. There's some people who still think that maybe it's always been there and now the frogs are more susceptible, but it's hard to believe given the genetic data. But that said, we still need to find some kind of explanation for why such an exquisite peptide has evolved. And I think part of the response is actually the combats. And so we tested whether some of the fragments were good in terms of healing wounds. And sure enough, yes, one of the two fragments, the function of that fragment is actually to speed up
the healing,
wound healing.
Yeah.
And so probably it's sexual selection that is driving the evolution of that peptide, at least of that fragment. It might be surprising that we basically have a peptide where the whole product is antimicrobial, and one fragment then has a completely different function, which is to help with wound healing. But not surprising if you look at other examples in nature. I mean, there's even viruses where their genetic code can be read both ways, and it's used in both directions to code for proteins. So it shouldn't be entirely surprising to see this kind of concentration of functions, right? Or use of the same DNA for multiple functions.
Very interesting species. Yeah. Could you tell us a little bit about your contribution to the book "Lost Frogs and Hot Snakes: Herpetologist Tales from the Field"?
Yes. That was an idea of Marty Crump, who's the author and editor of the book. She's published great books about natural history and about frogs and about frog decline. And the amphibian biodiversity crisis. And so she had this idea to basically collate all these stories from people that she knew and that she knew had spent a lot of time in the field pursuing herpetology. And so she asked me to contribute, and I was very happy to participate. And so my chapter actually was remembering parts of my PhD. And actually, my PhD was not with amphibians, was with desert communities that live on a landscape that kind of looks like the moon.
The Atacama?
The Atacama Desert, the northern Atacama Desert, which there are places there that have never recorded a single millimeter of rain. And so extremely, extremely arid. And so what is the source of productivity for all these desert organisms? And the response is that it's a coastal desert, and a lot of that a lot of those nutrients actually come from the ocean. And so for that, I was working in a peninsula, Paracas, in central Peru, and also on Guano Islands. I work extensively on those Guano Islands, where you have entire food webs, basically terrestrial communities that rely entirely just on what the ocean is producing. And you go from very tiny darkling beetles or solifuges, which are these very terrestrial desert organisms, the solifuges, actually eating beach hoppers, amphipods, up to condors. And the condors every year, they travel from the Andes, where they nest and they live. They spend most of the year. But every year between December and January, they fly down to the coast, because the sea lions is when they give birth. And so they're just eating all these placentas and there's a high mortality also of the pups every year in those massive colonies. And so the condors spend 2 months there. They eat, they gorge on all these placentas, and then they fly back to the Andes. And it's a connection that it can be quite important in terms of the input of nutrients. And again, with condors, it's going to travel over hundreds of kilometers. Obviously, with a solifuge, we're talking about a very small scale, spatially, but with other organisms, we're looking at much larger scales.
And what caused you to transition to herpetology then?
That's a very interesting question. I always, with that project also, I spent a lot of time working with geckos and lizards because I also work with scorpions and spiders, etc., arachnids, but basically any group that was easy to work with in the desert. I was still doing herpetology, just not amphibians. But no, I think I always had more of a passion for amphibians. But that specific project, when I was my first year of graduate school, I had many ideas about what I wanted to do and which questions I wanted to pursue. And I made a list, and then I basically circulated the list among the professors who I wanted in my committee, in my PhD committee, and asked them to rank the ideas. And so this one is an idea that consistently came at the top of what people preferred. And I think it also comes from being at Florida International University, where it's a strong focus traditionally on ecosystem ecology. A lot of people coming from Georgia with all this tradition in ecosystem ecology that they then applied to the study of the Everglades ecosystem. And so the ecosystem component perspective here comes because we're basically looking at how marine nutrients make their way into terrestrial ecosystems. And so some of the tools, like again, stable isotopes or fatty acid analysis, there are many tools that people also study in ecosystem ecology.
Let's talk about some of the challenges that are facing amphibians currently worldwide. I was looking at this paper here that you had written that state 40.7% of species are globally threatened. And extinctions continue to increase. Can you talk about what are the main threats to amphibians worldwide?
Yeah, I think a threat that's common to a lot of organisms is habitat loss, and we see that for amphibians as for many other groups. I think amphibians might be even more susceptible because many of them depend on both aquatic and terrestrial ecosystems. So sometimes the water part is not destroyed or altered, but the terrestrial part is, or vice versa. And that makes them even more vulnerable often to change in land use patterns. I will say that if you look just broadly, that's probably the biggest threat, and just developing the land or converting into agricultural, or just even development, just people building homes, etc. And then we also have all these emergent threats, which I think with habitat loss, you can, even though it's really difficult, but it is manageable to some degree. But some of those emergent threats, like disease, like these wildlife diseases, or like climate change, obviously they're much more challenging because it's something that it's very difficult to control. And we know it's difficult to control the spread of human disease. Imagine a microscopic wildlife disease, especially for wildlife like frogs frogs are, let's be honest, is not necessarily the most charismatic of vertebrates. Usually people think about mammals, they think about birds, then they think about maybe snakes or reptiles, and then it's frog, and then it's fish, etc. And it's insects. And so that poses challenges that I think are not unique to frogs. We see the case white nose syndrome with bats, but there's many fungal diseases also attacking plants and so on. But I don't think we can see any other example beyond amphibians where single disease has been so destructive in terms of driving diversity.
And I thought this was interesting too. Can you talk a little bit about the 3 most common breeding strategies for amphibians and how that can increase their risk of extinction?
Yeah, so amphibians among tetrapods by far have the greatest variety of reproductive modes. I say tetrapods because among vertebrates, fish also are incredibly diverse in the way they reproduce. But amphibians, certainly for terrestrial vertebrates, are the most diverse. And so I've already mentioned the marsupial frogs, for example. Now, that's not a common way of reproducing. Right. The most common is to have some type of aquatic larvae, and there is many variants. The species that often species just lay eggs in water, and then they hatch into tadpoles or larvae, and then you have metamorphosis, and then you have a terrestrial adult or sometimes an aquatic adult. But you have many variants where in some species the eggs are semi-terrestrial, or they're in foam nest, or they are hanging from leaves on top of the creek, or so there's many variations around that theme. And then also, in tropical areas, especially in the Andes, for example, what actually is also very common is terrestrial breeding. And the largest genus of vertebrates is Pristimantis, and it's a genus of terrestrial breeding frogs. We assume they are terrestrial breeding. We actually don't know for sure because we don't have the information on how they reproduce for each one of those more than 600 species of Pristimantis. But we assume they're all terrestrial breeding, and so that means the female will just lay eggs in a moist environment, not in water, and the embryo develop entirely within the egg, and they just hatch out and they hop away. Now, often they do receive parental care, and often the female will be there attending the clutch. In other species, the male. In other species, both sexes, and there's a lot of variation in how long they attend the clutches for.
And which one of those would be more prone to habitat loss or extinction? Which reproductive method?
Yeah, that's an interesting question. Species that require both environments, they'll be more sensitive, or they just have a higher chances if one of the two environment is destroyed, that then they cannot persist anymore. Now, that said, often there are the advantages to, for example, being able to be a tadpole. You can swim, right? You can swim, you can perhaps adjust better to variation, let's say, rising temperatures. You can swim from warm areas in the creek or in the pond. You can go deeper to cooler area. You're a mobile organism, you're a tadpole. If you think about, for example, terrestrial breeding species, the eggs are there, they cannot move. And often also many of the terrestrial breeding species, when you look at their physiology, they are less tolerant of extremes in temperature, for example, compared to many aquatic breeding species. There is a lot of variation. So it's not an easy question to answer, I think, because you have to take into account all these different variables that make it more or less likely for a population to persist. So I won't just focus on the exact timing of the development of the embryos.
Well, let's talk about some success stories as well. Could you talk about some amphibian populations that have rebounded or are showing growth?
Yeah, definitely. It's something that we have seen with this disease as should be expected, because what you typically see with a disease is that you have some type of epidemic spread, and eventually that wave ends. Even now, often you have multiple waves, but eventually you get to some kind of equilibrium. And so you would expect that once you reach that equilibrium, and if there has been some kind of selection for resistance or tolerance, and usually it's not so much resistance, which is not getting infected, it's more like tolerance, like the tree frogs, or being able to cope with being infected and keep it in check, then that's what you would expect. And that's what's been happening in several cases. And so the group, I think, that where those recoveries are most exciting are these harlequin frogs, which are these diurnal, highly charismatic frogs. They're actually toads. They belong to the family of toads. Some are bright yellow, red with black, green, etc. So, and often people know them because they are diurnal frogs. You can see them during the day. They're not, most frogs are nocturnal, but these ones are they just hop around, and usually they're just along creeks, and that's where people go to get water. So, for frogs, they can be quite charismatic, and people know about them, and people are aware of their presence. And so we also have fairly good data about when they started disappearing. and this group specifically is distributed from Costa Rica to Bolivia throughout the mountains of mostly the Andes, but also in Central America and all other mountain ranges. And so we have a pretty good idea of when many of those species declined or just kind of vanished. And at some point we had 75% of the genus, so 75% of about 120 species that people had not seen for 20, 30 years. And we basically were, they were kind of considered extinct at that point. But some of them now, over the last 5, 10 years, some of them have been rediscovered, which is great. Hopefully more of those rediscoveries, which also underscores the need that it's not because the species have not been seen that we need to stop protecting a habitat, right? We always need to make sure that habitat is there because species can't recover. But I fear that there's still a loss of genetic diversity, and these populations still basically underwent a genetic bottleneck that we know, just statistically speaking, is not good in terms of giving a long-term chance of persistence to populations, because, well, we know that the environment is constantly changing. With less genetic diversity, we're also looking at populations that might be less prepared to deal with some kind of massive change in the environment. But good news are always welcome. And so in the place where I've been working, I've already seen several of those species made recoveries. Initially, they were kind of timid recoveries. What I mean by that is I work in the cloud forest, and this site basically I've been going back for almost 30 years now. So I've seen how it was before, how it became a really sad place, and how it's now slightly recovering, although many species are still missing. But initially, the ones that made recoveries are species that have large population in the lowlands, and they're able to basically resettle those higher environments. Over the past 3-4 years, we've also rediscovered 2 species that are endemic to higher elevations, and one of them was just a kind of a very exciting, kind of emotional moment to hear it again, right after 25 years. Yes.
Oh, wow. Yeah.
So it's been a long time.
Do you think there's anything that we as humans can learn from amphibians?
Yes, I think amphibians are really great to study a lot of questions in biology. Traditionally, it's been that they are organisms that, perhaps that's not true of anymore for many species, but traditionally it's something that was very easy to study, and that's, I think, part of the reason why I study them is I never like to get up too early. I never like to not being able to handle the organism. I think there's a component of just being able to have them in my hands and examine them that really pushed me. I was never into trapping. Like mammals, you need to trap them. You need to set out traps and wait. With birds, you're basically observing them from far away with binoculars, or you're also trapping them with mist nets and so on. But with frogs, you just go out and capture them. And for being, again, a tetrapod, which is fairly similar to what we are, you can ask a lot of questions very easily because it's easy to get a good sample size. I think that we should really be concerned by the fact that they are so highly threatened, and again think about how similar they are to us, and again think about this disease specifically. Again, if you have a culture, a petri dish culture of lymphocyte, human lymphocyte, and you put the fungus there, it kills the lymphocytes, and so I think we have much to learn in terms of also resilience. Amphibians as a group, they have survived all these previous mass extinctions. The fact that right now they're in such trouble, I think is somewhat a little bit scary, right? And I think we can learn a lot from understanding specifically their resilience, what allows some of these species, for example, to persist despite this very catastrophic epidemic.
What do you think is the most important quality for a biologist to have?
Well, I think curiosity, really. I mean, I'm very much a field biologist. I always try to get out of my office and spend as much time as I can in nature. And what I really like about nature is you can, it's, I mean, a lot of it is somewhat predictable in the sense that you kind of know what to expect, but there's always an element of surprise. And usually that element of surprise is really kind of inspiration to ask novel questions. And I think it really satisfies this curiosity in ways that I think other activities don't allow me to. And it's a constant learning experience.
Yeah. What do you think was the best piece of advice that you've received over the course of your career?
That's a hard one because I mean, I think I've been lucky in having mentors that really valued creativity and asking questions and asking, I think, the right questions and critical questions, and just try to think that way and replicate that kind of approach is I think what has helped me a lot in my career. I think academic careers are not easy. They demand a lot of personal investment and sacrifice. And I think probably the best quality that I have definitely received, absorbed from my mentors has been this idea of basically constantly asking questions. Also, I think at the time it was pretty unusual, but basically most of my mentors have been women. In an academia, 30 years ago was not as common. And I think there's also an element of, again, resilience, of wanting to have your voice heard at a time when, again, that was pretty much a minority. I think that also helped in terms of finding your own voice and sticking with it and making sure that if you think that your line of action and your kind of research program is one that, again, satisfies your curiosity and that gives you the necessary confidence to pursue your investigations.
What was the most exciting or unique thing that you witnessed in the field?
I think anytime I see specifically something probably related to parental care in amphibians is like the birth of one of these tiny marsupial frogs is a special moment. That it's a unique moment. Now those are difficult, are typically difficult to be there exactly at that time. It takes a lot of time spent searching for these animals, and they're very discreet animals. But I would say those are very unique moments that moments that I cherish.
What is the last book you've read?
Gosh, now I'm actually don't remember the author, but I'm reading a book published by the Fondo de Cultura Económica in Mexico, and they have this series on scientific communication that are meant basically for a broad audience, and it's about extinction, and it's kind of a similar idea to the book Elizabeth Colbert published on extinction. It's a short book, right? That again is aimed at the broad public, but it's extremely well written. So that's what I've just read this past week. Really enjoyed, but sadly I don't remember the name of the author right now.
That's all right. Has there been a recent discovery in herpetology that's completely changed our understanding of these animals?
I think especially with novel technologies, there's constantly new information, especially coming from genomics, that is changing our way, the way we understand these organisms. And by having all this mass information from the genome, we can look for traits. We can also accumulate information that allows us to predict how they might respond, for example, to those threats. They help us understand the evolution of different lineages. And so I think it's pretty exciting to be alive at this moment where such massive amount of information becomes available. I think it's not unique to amphibians. It's something that we see across the biological sciences.
And with that being the case, where do you see research going in the next 5-10 years?
I mean, I'm very excited about the possibility of combining anything that basically is these type of very powerful molecular approaches to field research. They can complement each other in ways that make it a lot easier to get information that it can inform behavioral ecology, can inform conservation, can inform taxonomy. And I think just the lab work, if you want to call it that way, and computer work, because a lot of informatics alone is not sufficient, because you also need to know how that is relevant to the natural history of the organism. So I'm very excited. I'm trying to do that. For example, there's a lot of talking about the use of eDNA for conservation. And one of the ideas that we're pursuing right now is that we're working with this group of midges that specifically attack amphibians. They're called Coretralidae. It's a family of dipterans called frog-biting midges. And they are actually attracted by the call of the frogs, only the female. But what's interesting is the females, they require multiple blood meals from the frogs to be able to lay the eggs. That makes it very useful to basically now use, instead of filtering water, you could just trap the midges, for which you only need a speaker and a good call, a good frog call. And once you trap the midges, you can get the DNA of the frog out of the blood meal that they have. And so you can basically hire these midges to look for example, species that have not been seen in a long time, or to just get an idea about the frequency of different species of amphibians.
Have midge populations grown in relation to climate change?
So that's a very interesting question, and one of the questions that we're pursuing as well. It's not so much in response of climate change, although you would expect that, because first of all, they're nocturnal, and usually flying requires some kind of minimum temperature. So in insects below, 12 degrees Celsius, I don't remember what the temperature is, but they basically don't fly. And so the fact that we have rising temperatures most likely is really pushing these midges higher up along these mountain ranges. And they're really good vectors of diseases like trypanosoma, which in humans cause Chagas disease. So yes, I think so. Although what we are doing actually is to try to understand how these midges could have responded to the loss of frogs. And what we're doing is basically go out in the forest and do playbacks with the song of frogs that no longer exist at that site, and see what happens, see what kind of midges we find. And sure enough, I can anticipate you that some of the frogs that attract the most midges are the frogs that went missing, that are no longer there.
Mm-hmm. But we don't know if those particular species are extinct. They may just be not in that particular location.
Correct. And some of them might recover in the future. That's the hope. But at least we have not been hearing them for a long time. We also don't know if formerly there were more midge species that we don't know. It's a very understudied group globally. There's very few specialists worldwide in this group of midges. There's a lot of cryptic diversity. And they also don't seem to be that specialized. And so it's entirely possible that even though there are species of amphibians that might have gone extinct, frogs that might have gone extinct, that did not necessarily mean that midge species went extinct, because they can find other amphibian hosts. But again, we don't know. That's just the pattern we see, that in the greatest majority, and every time that people have looked at whether there are specialist species, the degree of specialization is not that deep. And those midges, they even are attracted to very different types of amphibian calls.
Have you had any dangerous or scary scenarios in the field?
Yes, one of the scary scenarios is what I describe in the book that Marley Crump led. and that was studying these terrestrial food webs on islands, on Guano islands. And we relied on fishermen to be able to take us to these different islands, cheaply basically. So we didn't have to hire a boat just to access the islands. And it was one of those trips where the fishermen never came back to pick us up. So, and that was a little bit scary because, again, we're in the Atacama. Those islands don't have water. We're surrounded by salt water, and it doesn't rain. So that was a little bit scary. Although, even though it's a very arid place, it's very foggy. And so luckily that island actually had a little peak, and so we just went up to the peak and condensed fog with plastic sheets, and that's how we were able to have water for the 2 days that it took us to be able to contact people at the reserve who then came and rescued us.
And did you find out what happened to the fishermen?
No, never found out what happened to the fishermen. Obviously, we never went back to that specific fisherman. But I think what happened is that place, so in Paracas, Paracas is a Quechua word that means basically a sandstorm. And so during the austral winter, it's usually the foggiest season, which helped us a lot because at least there was fog when we got stuck on the island. But it's also when you have those sandstorms. And those sandstorms usually the typical day in the Atacama winter is that the morning is foggy. And then you have the sun coming out around noon for a couple of hours, and then again it's foggy. And when you have that, you basically have, the ocean is very cold, right? Because of the upwelling of the Humboldt current. And because it's also foggy, because of that condensation, the desert never heats up, correct? So that's great. The fishermen probably would have picked us up, no problem. But when you have those Paracas, those sandstorms, usually at sunrise, you have a clear sky, and the sun starts heating up the desert. And so now all that heat is heating up the air, which lifts above the desert, which means all this mass of cold air now is moving from the ocean onto the desert, and that creates this massive, massive sandstorm where all this mass of cold air is picking up all the sand from the beach and just depositing, and literally even inside home you have layers of sand. So that morning that the fisherman was supposed to pick us up, the sunrise was one of those sunrises. It turns out that for whatever reason we still did not have a sandstorm that day, but probably the fisherman got up and saw this is not a good day to go out in the ocean, and that's what happened.
And how were you able to finally contact someone?
So that was 24 years ago, but I had one of these very early cell phones, which was more like a brick. Yeah. But those early models, right, they had these antennas, right, these retractable antennas that actually guaranteed some pretty good coverage because you could just get the antenna out and try to look for signal. And so we were able to basically call the reserve, and they were able to come because they have a boat. So they rescued us. But it was kind of a scary moment, I have to say, because you cannot live without water. So.
Right. Yeah. How has your work studying amphibians impacted your humanity?
I think, all my work is with people and with collaborators, and so it's always been a team effort. And again, it ends up really being a constant learning environment, and that's perhaps not directly from the frogs. It could happen with any other type of organism. But I think that spending time with people who have very different backgrounds from you, and working in the Amazon, most people, you end up working with people who have who come from completely different cultures, who have completely different type of life experiences, working with fishermen, working with students, etc. And I think being able to see reality, see the world from all these different perspectives, or at least see what people who have those perspectives tell you about it. I think that's part of appreciating humanity in ways that I don't know would have been possible otherwise. I mean, you can watch movies, you can read books, but I think, especially in terms of remembering and really learning, I think obviously being able to spend time and even just moving and learning at the same time, I think is something that really improves your ability to retain that knowledge and also help with becoming a more tolerant human being and more respectful and more appreciative, I will say actually, appreciative of all this diversity of perspectives.
Mm-hmm. What do you hope what would you like your legacy to be?
It's a general question, or?
Yeah.
No, because specifically right now, for example, I mean, I spend a lot of time working in this Guano National Park, serving these communities of frogs. And obviously, part of what I envision is that eventually I want other people to be able to pick up from I won't be able forever to go, and it's actually kind of an unforgiving environment. It's very steep, it's kind of dangerous, and so it's something that probably I can do maybe another time for the 30-year survey. Yeah. We are at a quarter century so far, and I've been in all of those. But that I would like for people to be able to continue that effort. And I think there's a lot of information that comes from these long-term data sets, and especially now with new technologies. We really need to have a good understanding of how it used to be, because of that problem of kind of shifting baseline, right? That people could get all this data now from, let's say, DNA or from camera traps, etc. But it's hard to put them in context of what used to be there, because we don't have similar data from 20 years ago. And the only data that we have is data that people collected in ways that are traditional survey methods that people can still do today and can still do in the future. And so I think that information becomes extremely valuable. And after that, I hope that mostly that my work motivates other people to pursue similar careers, similar kind of human experiences. I think it's very enriching in terms of finding a meaning for what you do every day, or just kind of what you want to do with your life. I think personally it's been something extremely enriching. I mean, I could die tomorrow and I'll be extremely happy with what I had and really the privilege and all the opportunities. I mean, I've seen things that have made me extremely happy, and I think that's something that I wish other people will also experience, and if that can be my legacy, I think it's something that I will find very, very valuable.
I think that's a great place to end. Alessandro, I appreciate the time.
Thank you.
Grazie.
Yeah, thanks.